Today we are going to start with the chapter number 3, Plant Kingdom of Biology, class 11th. So let's start. In the previous chapter, we looked at the broad classification of living organism under the system proposed by Whittaker, wherein he suggested the 5 kingdom classification with Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with the further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae. The earlier classification placed them in the same kingdom, so the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us look at the classification within angiosperm to understand some of the concerns that influence the classification systems. The earlier system of classification used only gross superficial morphological characters such as habit, color, name, shape of the leaves, etc. They were based mainly on vegetative characters or on the androecium structure. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems have given equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed which were based on natural affinities among the organisms and consider not only the external feature but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytochemistry, that is based on the cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. Now moving to algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic organisms. They occur in variety of habitats, moist stones, soils, and wood. Some of them also occur in association with fungi and animals. The form and size of algae is highly variable, ranging from colonial forms like volvox to filamentous forms like Eurothrix and Spirogyra. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated, similar in size as in Eulothrix, and non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous. Example, Volvox fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in the intermediate environment. They are of paramount importance as the primary producer of energy-rich compounds which form the basis of food cycle of all aquatic animals. Many species of Porphyra, laminaria, and sargassum are among 70 species of marine algae used as food. Certain marine brown and red algae produce a large amount of hydrocolloids, water-holding substances, example algin and carrageen, which are used commercially. One of the commercial products obtained from Gelidium and Glaciaria are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as a food supplement even by space travelers. The algae are divided mainly into Chlorophyceae, Phaeophyceae, and Rhodophyceae. The members of Chlorophyceae are commonly called green algae, and the plant bodies may be unicellular, colonial, or filamentous. They are usually grass green due to dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticular, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids, located in chloroplasts. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall, inner layer of cellulose and outer layer is of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated spores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells. It may be isogamous, aniso, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Pyrogyra, and Cara. Phaeophyceae. Members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous formed ectocarpus to profusely branched forms such as kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophyls. They vary in color from olive green to various shades depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrate, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contain, in addition to plastids, a centrally located vacuolar nucleus. The plant body is usually attached to substratum by holdfast and has a stalk called stipe. The leaf-like photosynthetic organ is frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most of the brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. The gametes are pyriform shaped, means pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with a great concentration found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at the great depth in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to myelopectin and glycogen in structure. Red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia. The common members are Polysiphonia, Porphyra, Glaciraria, and Geridium. Okay, now moving ahead to Bryophytes. Bryophytes include various mosses and liverbirds that are found commonly growing in most shady areas in the hills. Bryophytes are also called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks. The plant body of Bryophyte is more differentiated than that of algae. It is thallus-like, prostrate, or erect, attached to the substratum by a unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, stem-like structures. The main plant body of Bryophyte is haploid. It produces gametes, hence is called gametophyte. The sex organ in Bryophytes are multicellular. Male sex organ is antheridium. They produce biflagellate anthrozoids. The female sex organ is archegonium and is flask-shaped and produces single cell. The anthrozoids are released into water where they come in contact with archegonium, and anthrozoid fuses with the egg to produce zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called the sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nutrition from it. Some cells of the sporophyte undergo reduction division to produce haploid spore. These spores germinate to produce a gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of Spagenum moss provide peat that have long been used as a fuel and as packaging material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, they are the first organisms to colonize rocks and hence are of great economic importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mat on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverbirds and mosses. Liverbirds grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep into the wood. The plant body of a liverbird is thalloid, example Markentia. The thallus is dorsoventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverbirds takes place by fragmentation of thalli and by formation of specialized structures called gamete. Gamete are green, multicellular asexual birds which develop in small receptacles called gamete cups located on the thalli. The gamete become detached from the plant parent body and germinate to form new individuals. During sexual reproduction, the male and female sex organs are produced either on the same or on the different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule, so spores germinate to form free living gametophyte. Now mosses. The predominant stage of life cycle of a moss is gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from a spore. It is a creeping green branched and frequently filamentous stage. The second stage is a leafy stage, which develops from secondary protonema's lateral bud. They consist of upright slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular branched rhizode. This stage bear the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in secondary protonema. In sexual reproduction, the sex organs and 3D and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverbird. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of a spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Spagenum. Moving to for pteridophytes, include horsetail and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. Important. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into two roots, stem, and leaves. These organs possess well-differentiated vascular tissue. The leaves in pteridophytes are small, which are called microphylla as in Selaginella, and large macrophylla as in ferns. The sporophytes bear sporangia that are subtended by a leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called staurobilli or cones. The sporangia produce spores for meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular free living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical areas. The gametophytes bear female and male sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gamete released from antheridia to the mouth of archegonium. The fusion of male with egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of pteridophytes. In majority of pteridophytes, all the spores are of similar kind. Such plants, they are called homosporous. Genera like Selaginella, Salvinia, which produce two kinds of spores, macro and micro, they are known as heterosporous. Megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of zygote into young embryos take place within the female gametophyte. This event is a precursor to seed habit, considered as an important step in evolution. The pteridophytes are further classified into four classes. Psilopsida, Psilotum is the example. Lycopsida, Selaginella, Lycopodium, Synopsida, Equisetum, and Pteropsida, Diopteris, and Teres adiantum. Now moving for gymnosperms. The gymnosperms, gymnosnakids, spermacedes are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, they are naked. Gymnosperms, they include medium-sized trees or tall trees and shrubs. One of the gymnosperms, a giant redwood tree, Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others, cycas. Small specialized roots called colloid roots are associated with nitrogen fixing cyanobacteria. The stem are unbranched as in cycas or branched as in pinus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years, and leaves in gymnosperms, they are well adapted to withstand extreme temperatures, humidity, and wind. In conifers, the needle-like leaves, they reduce the surface area. The thick cuticle, sunken stomata, they also help to reduce water loss. These paragraphs are important. Gymnosperms, they are heterosporous. They produce haploid microspores and megaspore. Two kinds of spores are produced within the sporangia that are borne on sporophylls, which are arranged spirally along an axis to form a lax or a compact storobili or cones. The storobili bearing microsporophylls and microsporangia are called microsporangiate or male storobili. The microspores develop into male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cone bearing megasporophylls with ovules or megasporangia, they are called macrosporangiate or female storobili. The male or female cones or storobili may be borne on the same tree, Pinus. However, in cycas, the male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the other cells of the nucellus. The nucellus is protected by envelopes and other composite structures. Is called an ovule. The ovule are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within a megasporangium, they develop into multicellular female gametophyte that bears 2 or more archegonia or female sex organ. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovule and discharge their contents near the mouth of the archegonium. Following the fertilization, zygote develops into an embryo and the ovules into the seed. These seeds are not covers. These seeds are naked, because they do not have the covering. Now moving ahead to angiosperms. Let me change the color. Unlike gymnosperms, where the ovules are naked, the angiosperms are flowering plants. The pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in the fruit. The angiosperms are an exceptionally large group of plants occurring in the wide range of habitats. They range in the size from smallest Wolfia to tall trees, Eucalyptus, over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes: dicots and monocots. Dicots 1 cotyledon, dicots 2 cotyledons, and monocots 1 cotyledons. Dicot have reticulate venation, monocots have parallel venation. Now this is all about this chapter. This is the diagrams, and for NEET some topics are missing, like diplontic phase, haplontic phase, and haplodiplontic phase. These are missing. We will try to cover them too. ठीक है, तो haplodiplontic phase is a phase in which half of the plant growth period is haploid. That is, wait, haploid means gametophyte, and diploid means gametophyte. ठीक है, and diploid means sporophyte, means 2n condition. Yes, so in gametophyte this is n condition. Mitosis will occur. In sporophyte condition, meiosis will occur, as it is 2n. And diplontic examples are gymnosperms, angiosperms, and fucus. And example of haplontic life cycle, they are algae like volvox. ठीक है, but haplodiplontic example is Actocarpus. Okay, and bryophytes, all the bryophytes, pteridophytes,